Hi everyone, in this lecture we will be talking about cell signaling. Just as humans need to communicate with each other in order to form a stable society, the cells in a multicellular organism also have to talk to one another in order to ensure the proper function of the organism. And this is where cell signaling comes into play. The other name for cell signaling is also known as signal transduction, and this is because in cell signaling it mainly involves the conversion of signals. So cells have to transmit signals, and they also have to receive signals and convert those into something that the rest of the cell can understand. There are two types of cells involved in cell signaling. The first one is called the signaling cell, and this is the cell that creates the extracellular signaling molecules. Another cell receives this molecule, which we call the target cell. It detects the signal molecule using a receptor, and it then creates an intracellular signaling molecule that the rest of the cell can understand. So here you can see an example of the conversion at work. Here the cell is converting the extracellular signal into an intracellular signaling molecule. Now there are different types of extracellular signaling. The first one is called endocrine signaling. Here, signaling cells send signals throughout the whole body in the form of hormones. You can think of endocrine signaling as a long-distance signaling that occurs all throughout the body. And it does this by transmitting these hormones through the bloodstream. So any cell that can be touched by your bloodstream is a potential target for your endocrine signaling. The next type is called paracrine signaling. Unlike endocrine signaling, which broadcasts its signals all throughout the body, paracrine signals only signal within the local area. And it does this with the use of local mediators. So here you can see the signaling cell and the local mediators it produces and the different cells around it that detect those local mediators. There is also a special type of paracrine signaling which we call autocrine signaling. In this type of signaling, the signaling cell is also the target cell. So it produces the different local mediators, and it is also the cell that is detecting those mediators. In this case, we can classify this type of cell as self-stimulating. The next type of extracellular signaling is called neuronal signaling. Just like endocrine signaling, signals in this pathway can also travel long distances. But instead of using the bloodstream to transmit the extracellular signals, it instead uses specialized cells called neurons. Here we have an example of a neuron. The neuron has a very long tendril which we call the axon, and in some cells this can be as long as 1 meter in length. While signaling in the endocrine can have a broad range of target cells, neuronal signaling is more rapid and specific. This is because the signaling involves direct communication between one neuron and one target cell. In between these two cells, there is a gap called a synapse, and this is where the extracellular signal, in this case called a neurotransmitter, passes through. The last type of extracellular signaling pathway is called contact-dependent signaling. As the name suggests, this depends on direct physical contact between the signaling cell and the target cell. The signaling cell has the extracellular membrane-bound signal molecule attached to its cell membrane, and in turn the target cell also has a receptor attached to its membrane. And together, they both have to make contact with each other in order for the signal to be transmitted between the two. Next, let's talk about the target cell response. Signals can cause different responses depending on the target cell type, and this is all based on the presence of effector proteins in a variety of cells. These proteins are the ones that have a direct effect on the behavior of the target cell. In this example, we can see the extracellular signal acetylcholine being detected by 3 types of cells. We have heart pacemaker cells, salivary gland cells, and skeletal muscle cells. And even though the same signal is being detected by all 3 cells, they have different responses. In the case of the heart pacemaker cells, there is a decreased rate of firing, which leads to a slower heartbeat. In salivary gland cells, there is an increased secretion of saliva, and for skeletal muscle cells, there is contraction. So the reason for these different responses is due to the different effector proteins in these different cells. Next, the target response also depends on the combination of signals received. In this example, we can see the same cell receive 3 signals, A, B, and C. But 2 of these cells receive another set of signals. This cell gets your D and E signal, and this other cell receives the F and G signal. And you can see that even though all 3 of them have the A, B, and C signal, these 2 have a different response because they have another set of signals. In this case, sometimes when cells don't receive any signals, then they are triggered to do apoptosis or cell suicide. The target cell response can also have a variety of speeds. This is largely dependent on the availability of effector proteins in the cell. In this example, we can see 2 types of responses, one fast response and a slow response. In cases where the effector protein is already present in the cell, the response can be very rapid, ranging from a few seconds to a few minutes. Meanwhile, if the protein is not yet present in the cell, the signal needs to travel to the nucleus so that the gene for the protein in question can be expressed, and then the gene needs to be created before the cell can even produce a response to the signal. In this case, the response can take as long as a few hours to be seen. Next, let's talk about our different receptors. These are proteins that are activated by a specific signaling molecule. And we have 2 classes of receptors basing on where they are located in the cell. The first class are called cell surface receptors, and as their name suggests, they are located on the surface of our cell. These receptors are used to detect extracellular signaling molecules that are too large or that are not able to enter the cell. In these cases, the cell surface receptor protein has to convert the extracellular signaling molecule into an intracellular signaling molecule so that the rest of the cell will know how to respond. On the other hand, we also have intracellular receptors, which are often called nuclear receptors. These are receptors that are found in the cell, and many of them are found in the nucleus, and they are used to detect small hydrophobic signaling molecules which can readily pass the cell membrane. In this lecture, we will be focusing more on cell surface receptors. Again, these are receptors that recognize the extracellular signal and generate a new intracellular signal in response. In this figure, we can see the function of our receptor. We can see that it has already detected the extracellular signaling molecule, and now it is this receptor's job to convert the extracellular signal into an intracellular signaling molecule in the process called transduction. Sometimes the extracellular signaling molecule can just be one involved, but most of the time many molecules are involved and they form a pathway like this. The signal becomes converted from one molecule to the other to another until it reaches the different effector proteins of our cell. And at this point, this is the time where we can see the target cell response. Moving on deeper into the intracellular signaling pathways, these pathways have a variety of functions. The first one is simply to relay the signal. Here we can see that the relaying ability of the pathway just passes the signal from one protein to another and then to another. Afterwards, the pathway can also amplify a signal, meaning it can take a simple signal and multiply it to make sure that it is well received by the cell. Next, there can also be an integration or a combination of signals. Earlier we mentioned that certain cells have different responses depending on the combinations of signals they receive. So some proteins in this pathway combine different signals and change them into a new signal. Next, the pathways also distribute the signal to the different effector proteins. And lastly, and most importantly, these types of pathways provide a feedback to the rest of the cell. Now, the feedback tells the cell to keep going with the pathway or to stop. In all biological systems, the presence of feedback loops is essential for proper function. We'll be talking about 2 types of feedback loops. The first one is called a positive feedback. And in this feedback, we can see that the cell's response is enhanced. In this example, we have two proteins involved in a signaling pathway, protein T and protein Y. The presence of protein T stimulates the production of protein Y, and once protein Y is present, it talks back or gives feedback to this pathway to continue creating protein T, or it can also increase the production of protein T, and this leads to the production of more protein Y. On the opposite side, we also have a negative feedback pathway, and the response of the cell in this kind of feedback is diminished. So going back to our two proteins, protein T and protein Y, here we can see that the production of protein T stimulates the production of protein Y, but once protein Y has been produced, it talks back to this pathway to either stop the production of protein T or to diminish the production of protein T. Feedback loops in the cell either turn on something in the cell or turn something off, and those loops rely heavily on molecular switches. These switches are incredibly important. In fact, this is actually the most common function of intracellular signaling molecules. There are two possible actions for the molecular switch. One is to stimulate the cell or the pathway, and the other one is to suppress the cell or to suppress certain pathways. Molecular switches are incredibly important because each activation step in the cell needs to have an inactivation mechanism. So anything in the cell that can be turned on needs to be turned off eventually. We will now be talking about the different types of molecular switches in the cell. The first ones are the proteins that are activated and inactivated by phosphorylation, and this one is the most common type of molecular switch in the cell. Here we have an example of this protein switch. Here we can see its off configuration and its on configuration. The way that you turn this protein on is by phosphorylation or the addition of phosphate. Two other proteins are responsible for controlling these types of switches, and the first one is called protein kinase. This is the protein that attaches a phosphate group to our molecular switch. It takes this phosphate by hydrolyzing ATP into ADP. The next protein is called protein phosphatase, and it does the opposite. Instead of attaching a phosphate group, it takes off a phosphate group again. By taking off the phosphate group, we are inactivating the molecular switch. The cell can also contain phosphorylation cascades, and these are chain reactions that occur when the activated protein or the activated molecular switch is also a protein kinase. So once this switch is turned on, it can then stimulate another switch to turn on through phosphorylation, and then another and another. There are two types of protein kinases. The first one is called serine-threonine kinases. This one is the most common type of kinase. We classify kinases based on where they attach the phosphate group to, and in the case of serine-threonine kinases, they attach the phosphate to either a serine or a threonine amino acid. The second type of protein kinase is called tyrosine kinases, and instead they attach the phosphate to an amino acid called tyrosine. The second type of molecular switches are called GTP binding proteins. Instead of relying on phosphate to turn these proteins on, they instead rely on the compound called GTP or guanosine triphosphate. In the active state, these proteins have GTP attached, and in the inactive state, they instead have another compound called GDP or guanosine diphosphate attached. One thing that is unique to these proteins is that they have an intrinsic GTP hydrolyzing or GTPase activity. This means that it can turn itself off or do an auto shut off by hydrolyzing their bound GTP into GDP. So in this figure, we can see the GTP being hydrolyzed and being converted into GDP by the hydrolysis or the removal of phosphate. Now there are two types of GTP binding proteins. The first one are called monomeric GTPases. Now these monomeric GTPases are only composed of one protein subunit, that's why they are called a monomer, and they need the help of certain regulators in order to function. The first one is called guanine nucleotide exchange factors, or GEFs for short. And these GEFs promote the exchange of GDP, that means they promote the removal of GDP, and the exchange for GDP. On the opposite side, we also have the GTPase activating protein, or GAPs for short, and these proteins promote GTP hydrolysis. In this figure, we can see the effects of these regulators. Here we can see GEF and its ability to turn on our molecular switch, and our GAP and its ability to turn off the molecular switch. The next type of GTP binding proteins are called trimeric GTP binding proteins. This can be also known for its other name, G proteins. As the name suggests, it is trimeric. That means it is composed of 3 protein subunits: the alpha, beta, and the gamma subunits. In this figure, we can see the attachment point of the G protein to the cell membrane. Here we can see that the alpha subunit is attached to the cell membrane, as well as the gamma subunit. Meanwhile, the beta subunit is attached to the gamma subunit. Let's now move on to the different types of cell surface receptors. The first class of receptors are called ion channel coupled receptors. And as the name suggests, they are an ion channel and a receptor at the same time. So here we can see the channel in its closed state when it cannot detect the extracellular signaling molecule, but when it does detect the signal molecule, it opens up and it allows ions to enter the cell. And this alters the membrane potential of our cell. The second type of cell surface receptors are called G protein coupled receptors, and their main function is to activate G proteins. This set of receptors make up the largest family of cell surface receptors and the most diverse, but they have a very similar structure for all of them, in which the protein passes through the membrane 7 times. So here we can see that it passes 1, 2, 3, 4, 5, 6, and 7 times throughout our cell membrane. This structure allows it to have 2 main domains, one on the outside or the extracellular surface, which is used to detect the signaling molecule, and a domain on the inside which is used to activate the G protein. In this figure, we can see how the G protein coupled receptor activates the G protein. So here we can see the 3 inactive parts of this pathway: the inactive receptor, inactive G protein, and an inactive enzyme. Once the receptor detects the signaling molecule, it then binds to the G protein and then activates it. And in turn, the G protein can activate the enzyme or another type of pathway inside the cell. Let's take a closer look at the activation of our G proteins. Here we can see the inactive receptor protein and the inactive G protein. In the inactive state, the G protein has 2 distinct parts. We have the alpha subunit, and while there is GDP attached, it is inactive. Meanwhile, the other 2 subunits, which we call the beta-gamma complex, remain inactive so long as they are attached to the alpha subunit. In order to activate, the GDP of the alpha subunit has to dissociate and it needs to be replaced by GTP. Meanwhile, for the beta-gamma complex, they become active once the alpha subunit has detached from it. And normally this happens right after GTP has attached to our alpha subunit. Now these different subunits can act directly on other enzymes or other parts of the pathway in the cell. Next, let's talk about the inactivation of our G proteins. Our G proteins again have 2 distinct parts, the alpha subunit and the beta-gamma complex. For the alpha subunit, it also has an intrinsic GTPase activity just like the monomeric G binding protein. So after a while, it can convert GTP back into GDP and inactivate itself. When this happens, the 3 units reassemble, which we can see in this portion of the figure. This occurs within a few seconds after activation, and what this does essentially is that it shuts off whatever pathway these 2 components have triggered. Next, let's talk about the different G protein targets. As we mentioned earlier, once the G proteins become activated, they in turn can turn on other proteins in the pathway, or can affect other parts of the cell. The first target of our G proteins are ion channels. And these ion channels cause immediate changes in the cell because once they open, they allow ions to enter the cell or exit the cell and leads to changes in the membrane potential of the cell. So here we have an example of a potassium channel which is still closed, and it is activated by the beta-gamma complex of our G protein. Once the alpha subunit becomes inactivated, the three subunits reassemble and the beta-gamma subunit detaches from our potassium channel. In doing so, it also inactivates the potassium channel by closing it. The next type of G protein target are membrane-bound enzymes. Now, these are proteins that catalyze a variety of reactions in the cell. And unlike your ion channels, these enzymes produce a response that is less rapid. When they are activated by the G protein subunit in charge of them, this leads to the production of additional intracellular signaling molecules called second messengers. One example of a very important second messenger is cyclical AMP. We will not delve deeper into these different second messengers, but please look forward to another lecture on those. Now let's talk about the last type of cell surface receptor, which we call enzyme-coupled receptors. In the previous slide, we needed the G protein to activate our different enzymes. But for these receptors, they themselves can be the enzyme, or they can activate an associated enzyme directly without the need of our G protein. Most enzyme-coupled receptors are dimers, meaning there are two parts that have to come together in order to activate the enzyme. Here in this example, we can see a dimer, which is also an enzyme. And this extracellular signaling molecule is combining those two dimers. So when they get close together, this activates their catalytic domains. On this side of the figure, we can see a signaling molecule that is bringing together the receptor, but this receptor, instead of activating itself, it activates an associated enzyme. And this enzyme in turn can create the second messengers, which we talked about previously. Lastly, let's talk about different substances that can affect our receptors. Here we have some examples, and some of them include drugs, and others include everyday compounds that we encounter in our food. They can have two possible actions on the receptor. The first one is either to stimulate the receptor, as is the case with your barbiturates and your benzodiazepines. And you also have some that block certain receptors. Let's take, for example, capsaicin, which we can find very commonly in chilies and other spicy food. They stimulate the temperature-sensitive ion channel coupled receptors, which is responsible for detecting whenever we eat something that is hot. So when we have capsaicin in our food, even though the food is not hot, we are actually tricking these different receptors into sending a signal back to our brain that what we are eating is hot. And have you noticed that when you eat something that is hot and is also spicy, this actually doubles the reaction and makes the food even hotter? Alright, so this ends our lecture. If you would like to learn more about the things we discussed, please check out these two references. Thank you for listening, and we hope to see you in the next video.